Right. So once again, I am very thankful for the opportunity of doing this interview via Zoom. And as I was talking to Nathan a few minutes before, I have been extremely happy and extremely grateful for the quality of the questions raised. So today I prepared something that I'm going to do on the board with a little mathematical derivation, but it's just supposed to be the first part. And after that, we can talk about, some of projects that I want to develop within ICTP. And I think that this one that I'm working on, I'm going to present the mathematics behind a paper that I published, and I made a comment on the before about obtaining maximum information from Petri dishes. Now, before talking about this, I would like to know if you guys are familiar with this kind of object.
Yes.
Right? These are six-sided dice, right? So you imagine that when you roll one of these, you have these numbers from 1 to 6. And when you roll it, that is an exact equal probability of landing in whichever one of these numbers. Now, I have a bag here full of these D6s, and I'm going to roll them here. Now, what I'm going to tell you is, by this rolling here, I roll 2 times the number 1. Now, the question is, how many dice were in the bag? If I roll 2 ones, how many dice were in the bag? Well, many people would, from a question like this, come up with the direct answer. Oh, okay. Each die can have a 1 with a probability 1/6, right? So in every 6 dice, you should see 1, 1. So if you saw 2, you should have 12 dice. Now, of course, anybody might give this answer, but it could have been 13, it could have been 14, it could have been 50, right? In principle, none of these is unreasonable or beyond the beyond the possibilities of this experiment, right? Now, one of the first things that we learn from probability theory, it is to say, oh, I actually have an answer to this, right? So if I call K the number of ones, and then N the number of dice, I just want to confirm everything. You guys can read as well?
Yes.
Okay. And then you might say that the probability of K given N is going to be equal to N factorial, N minus K factorial, K factorial. So this is how many combinations you can have of K1s within N dice, and then 1/6 to the K, and then 1 minus 1/6 to the N minus K. Right now, to be completely clear, this piece here is the probability that I get K1s. This is the probability that I get N minus K, not once. And this is how many combinations of these can I get. Now, in the end, this question here is the answer to a different question, right? It's saying, if I roll 12 dice, what is the probability that I get 2 ones? What we're interested in here is the other way around. Base that I rolled and I got 2 ones, how many dice were in the bag? Now, to do this, to answer this question, we go to something called the basis theorem, which is a general result of the theory of probabilities, and is going to tell us that the probability of A given B in general, so if I have 2 events, A and B, is going to be equal to the probability of B given A times the probability of A divided by the probability of B, right? And I made that comment a little bit yesterday, but the idea is, oh, if I have some belief, if I had like some plausible belief on A, and then I have some kind of I have the result of my experiment. How do I update my belief in A? So on this case, this is quite straightforward, right? So here, A is going to be our parameters, in this case, N, and B is going to be, in this case, our data, in this case, the number of ones, K. So the probability of N given K is going to be equal to the probability of K given N, which is what we have from the binomial, some prior probability of N divided by the total probability of K. Now, the total probability of K is going to act more or less like the partition function in statistical physics. Now, many people asked questions yesterday about how to do hypothesis testing, and the probability of K in that case is going to be important. But like, if I want to just learn about N, if I just want to learn about the number of dice in the bag, what I'm going to do is to, I can ignore these and then just put these as a proportionality constant, right? And then know that in the end, probabilities have to sum to 1. So what I would have is that the probability of N divided by K is going to be equal to exactly what I had on the left-hand side, N minus K, K factorial, and then 1/6 to the K, 5/6 to the N minus K. Now, for the prior, we don't want to bias out of this. Let's just say that, like, if I can hold the bag of dice in one hand, it was smaller than, let's say, 100, right? So I'm just going to put like 1 over N max. So you create an N max that you say like larger than this N max. You attribute a prior probability 0. And what you have is this formula here, and you're going to have like some kind of proportionality constant here. And you decide what your proportionality constant is by summing over all possible values of N up to N max and saying that it has to sum to 1. Now, when you do this, the result that you get from this number is going to be something that peaks at the 12, which is exactly that number that we thought about. But it's also going to give you, oh, what is the probability that it was a 14? What is the probability that it was a 15? What is the probability that it was a 40? And what gives us is that, like, it's not only it's going to give you an answer, it's going to give you, like, could be this answer, could be this slightly different answer, could be that other answer, right? Now, the way that we usually work to say, I don't have an answer, but in principle, if you run the experiment multiple times, in this case, if I roll the dice over and over again, I would expect the probability to sharpen. How do we do that? Well, it happens that you say that I have multiple experiments. So I rolled and I got 2 1s, and then I roll again, I got 3 1s, 4 1s. And then I say that the probability of the set of Ki given N is going to be the product over I of P of Ki given N. Now, when you do this, from the same bag of dice, this is going to happen. It's going to get sharper and sharper. And you can say that at some point you create a cutoff. Usually people like the 0.95. So if one of these ends get a probability that is larger than 0.95, you say that you found the right number. And there is a lot of controversy about this. And there are 2 big things that you take from this. The first one is that not every time, I'm just rolling dice, not every time you can have exactly the amount of data that you want. And when you do this, you actually are doing something that is more complete, that you actually get, a quantifying uncertainty on you get based on your measurements. Now, I don't know if it's going to be surprising or not, but the number of dice that were rolled were 20. Now, a more interesting question, I guess, it is saying, oh, this is a position in biophysics. The title say Petri dishes. What does this have to do with Petri dishes? Well, the way that we count bacteria has the same mathematical structure as this problem. And that's what I'm going over right now. So,
sorry, maybe I could interrupt for just a moment.
Come again? Yes.
I had one question. So you didn't say anything wrong, as far as I can tell, but the fact that you rolled 2 1s, sometimes in a situation like this, I should maybe take that what I observed is that you rolled 2 things that were the same, and the rest were different.
Yes.
And so I don't know what, you've taken seriously the fact that they were 1s, and you haven't told us anything about the others.
Yes, exactly.
It's a very literal description, right? And I don't know whether this will be important in the problem you really do.
It will, because what I'm going to say is that this has a lot to do with the way that we measure bacteria.
Okay.
Right?
Okay.
Because I could have given you the 1s, the 2s, the 3s up to the 6s, and then you could just sum them and say, oh, I have my answer there. Correct?
Well, but also you could have said that I saw 1 coincidence.
Yeah.
Right.
And these would be more information, and maybe
right.
you'd have the probabilities based on that.
Good. Okay.
Now, why am I talking this like about giving just the 1s? Is that the way that we usually, I don't know how many of you are familiar with Petri dishes, but Petri dishes are objects, right? Are circular objects that they have some nutrients there. And the idea is that if a bacteria falls into this Petri dish, it's going to generate a colony. So this could give you an idea on how to quantify bacteria. I could go outside and I want to know, oh, how many bacteria are in a sample of my garden. I take a little sample of my garden, I take a vial, and then what I'm going to get is like I am going to mix up my vial and then put it on the Petri dish. Now, if I do that, I'm actually going to get something like this. You're going to have so many bacteria in general that you're not going to be able to distinguish single individual bacteria anymore. You're not going to be able to distinguish individual colonies. So what microbiologists usually do is to say, I get my vial, and I am going to dilute into a larger volume. And I'm going to take a subsample of these, and then I am going to put it in the Petri dish. Now, the dilution not necessarily have to be just 1 step like I have here. Usually it's made on multiple steps where you have like, 1 microliter into 10 microliters, so on and so forth. But the idea is, let's call phi total dilution factor. So this means how much of the original sample is in the Petri dish. Now, coming back to Professor Biliak's question, this is my 1s, right? So I'm not going to be able to see how many were outside of that. I'm only going to be able to see the 1s or the ones that were subsampled. Does that make sense? Yeah, excellent. So many people did exactly the thing about saying, oh, if I roll 2 1s, it's a 12, right? So throughout many papers in bacteria ecology, people just say that the number of bacteria is going to be K, the number of colonies that I observe, times the total dilution factor. Now, if we do, and that is something that is relatively easy to implement, which is, I am going to simulate this experiment, right? I get an N that I sample, let's say from a normal, with a specific mean and sigma, you make sure that like after that you use this as an integer, and then you sample k, the number of colonies on a binomial, which is the same n, and 1 over 5. So in this case, coming back to the rolling of dice, you have your number of bacteria is the how many dice you roll, and 1 over 5 is the probability that you roll the 1. So 5 would be the total number of faces that you can pick up. Now, if you do this, and you're going to see that as you do this, where you calculate k and then you multiply by 5, you're going to get one distribution, but the real distribution is a lot sharper. Right? So the idea is the real distribution of n is going to be this thing that I have in white, and the distribution that you obtain by just multiplying it by the dilution factor is what I have here in green. So I created a method to solve that, and that is called repop. And it was recently published at eLive. And what repop does is that you give your table with your number of counts and your respective dilutions. And it's going to give you a distribution here, P of n. Right? Now, one thing that is important to talk about this is that in this case, P of n also represents the fact that your samples are heterogeneous. Right? So like it might be that depending on when you are going to pick up your sample from the soil or from the ground or for whatever object that you're trying to study, you're going to have a non-homogeneous set. It's not like exactly like the dice. It's not. I'm always rolling the dice from the same bag. In this case, it would be like, oh, what if I roll the dice from a different bag that was produced under a certain structure? So many people look at this and say, oh, this is the heterogeneity of my samples, right? And something that you miss, and I think that is quite important to bring up, is that sometimes biologically, you might have multimodality, right? So it might be multimodality in white, and it might be that like when you only do it in the naive way, you're going to lose this multimodality. And the repop also is able to capture that. So there is intrinsic biologic information that you are missing when you don't take the binomial probability into account. Now, to give us an idea of how I'm able to achieve that, it's probably erase it all. What I do is, I'm going to say that the probability of n is going to be some probability of n given a certain set of parameters theta. Now, for repop, what I did is to use a mixture of Gaussians. So this is going to be 1 over c constant, and then a summation of rho alpha x of minus n minus mu alpha over sigma alpha squared, and then 1/2 alpha. And what repop does is to learn what my sigma is going to be equal to these rhos, mu's, and sigma. So the mean, the standard deviation, and the weights of every Gaussian. Right? So this is done this way. So I want to say, what is the probability of theta? What are the probability of these three, given my set of ki's? And this is going to be proportional to the probability of all the KIs given theta plus times your prior on theta. This piece here is what you're interested in. This one you usually try to keep so that this is as broad as possible while maintaining that you prefer fewer components. Now, when you do this piece here in green, it's going to be separated in a certain way. Now, we think about the measurement, and the biology is going to tell us how your parameter theta gives you the number of bacteria. And then the number of bacteria is going to inform your counts in the plate. So a way that we write this, that we call a graphical model in probabilities, it is to say, I go from theta, my parameters, to n, to k. And in this case, if I have multiple replicates, I'm going to call it ni, n1, k1, and then it's also going to inform n2, that informs k2, and then so on and so forth until n, let's call it big I to k, big I. Now, this term in green, all of this you should say, can be separated into the probability of each ki product over I of the probability of each ki given n times the probability of n given theta. And you have to integrate, or sum in this case, because you're dealing with something discrete, over all possible values of n up to your n max. So you calculate this here with your binomial, like I explained before, and this here with your Gaussian mixture. Now, what repop does is to find theta that is going to be theta star, that is going to be the argument maximum of P of theta even out of Ki over theta. So I take all my data and then I say, oh, what is the best parameters of my Gaussian mixture model that explain this K? Does that make sense?
Can you still tell us when this is a good approximation and when this is a bad approximation?
I mean, this is in principle the physics of the measurement, right? Oh, you mean the Gaussian mixture?
Yes.
Yeah. Many times people just use the Gaussian mixture as if you don't know what the format of the distribution is, you toss the Gaussian mixture. That's why on repop I use the Gaussian mixture. However, and that's part of the point, I believe that we can get this probability of n given theta. We shouldn't use the Gaussian mixture if we have a model for the system. So this is probably what most people are going to accept when you don't have a model for what the ecology of the system is. Now, when you have a model for the ecology of the system, that's where, and that's part of the projects that I'm proposing.
Okay. Can you give an example, for example, of a situation where it just doesn't work, this Gaussian approximation?
That's exactly what I was going to follow up to.
Okay, so then, yeah, please go ahead.
Yeah, so if it's all clear here, it's just one more thing that I want to say here is that this piece here, right, is the stochasticity in your measurement. Now, this is the stochasticity of your ecological dynamics. And then I have to marginalize over all of them. Now, for me to be able to find the argmax, I used a very nice tool called AutoGrad. In my blog, I have a few explanations of how AutoGrad works, but it's a way that, like, when you plug your function on Python or PyTorch, it's also going to give you a way that is also going to calculate the derivatives. So optimization problems are very facilitated by this autograd feature. Also, autograd is what made neural networks training possible, right? Because imagine when you have like these complicated functions that we have in neural networks if autograd were not there. But anyway, so coming back exactly to Nathan's question, like, oh, to what point can we trust the Gaussian mixture? And the point is like, in general, not. So I'm going to give one example that I believe it's what a model that is going to be very good for example, the project on the formation of biofilms that I was suggesting, right? So the idea is you have this bacteria arriving, and they eventually might attach to a surface. Now, these I can represent as a, let's say, attachment of bacteria with a rate alpha. Now, in the films of attachment, this is known to be a very rare event, right? So, for example, if we talk about microfluidics devices, this is the order of 1 in every 20 hours, right? So then you might say that your B is going to reproduce. So you have your bacterium attached, and it's going to generate more. And sometimes your bacteria is going to leave the biofilm, right? So I can represent these as B to the n. And I can call these, let's say, mu and k. Now, what happens in distributions like this is that we are going to have something that is quite similar to the dB number of bacteria. dt is going to follow a process that is similar to a logistic equation, right? So I could rewrite this to say mu minus mu over capacity, defining capacity as mu over k, times b times b. However, a differential equation like this is not going to capture everything, right? This is just going to give us an expected value. And for problems like this is not so good. Like if I simulate exactly this system here, with these rare events, what I'm going to observe is that my possible distributions, even though they are going to achieve capacity, they are not going to concentrate around one. So if I just say something like this, I will just get like this one in green, which is not the same thing. So with the things that I developed yesterday, we can come up with a good form to obtain the probability of the number of bacteria in time. And in these rare event cases, what happens is that this is quite bimodal. But in general, whenever we're working with these ecological models, you're going to have to come up with what are the important things on your model instead of something as generic as the Gaussian mixture. Does that answer your question, Nathan?
Yeah, I think so.
Yeah, right. So the point is like you have to model what are the relevant mechanisms behind the system that you're trying to study, and that I want to work on formation of biofilms. I already have a similar work on gut microbiota, right, where you have the experimentalists cultivate some microscopic worm, and then you try to learn what is the number of bacteria inside the gut of the worm. Yeah. So the important thing here is that then what is going to give this now, right? As I said before, the probability of n given your parameters is going to be given by your ecology model. Does that make sense?
Sorry,
Excellent.
so if I think about the example where you're sampling the microbiome of C. elegans,
Mhm.
I think of the different observation. I mean, so you started with an example where I have one big vat and I'm diluting it, right? And so
Yes.
the probabilistic description of the vat seems, I mean, that seems like it's reasonably deterministic. But then if I am sampling from the gut of worms, the different worms are different from each other in a way whose distribution could be very complicated because they have different experiences, they have different.
But yeah, that's exactly what the point that I'm bringing. Because what I'm saying is that when you pick up the C. elegans, you have to crush it, and then to learn how many bacteria are inside, you do the dilution.
Mm-hmm.
And that's exactly my point. Like coming back to the question that I had, to the equation that I had here, is that the probability, right, of your counts given theta, and this theta here, you can think about the parameters of your ecology, is going to be equal to the sum over n. Let's just say this for just a single k, right? Of the probability of k given n, probability of n given theta. Now, what the study of the vat gives us is this piece here, right? So like if I have a worm and it has in its gut 12 million bacteria, what is the probability that after I do the dilution, I obtain a plate that has 400 colonies? This part, it's already done in my previous paper. And it's something that like it was not being observed for a long time. Now this one, and you said it and you're correct, is going to depend on the ecological model. Right? And on this case, I am going to get, and I could come up with simulations from this, but on the model that I had written there, you do get like something like this. So here is your capacity, it's going to fluctuate around the capacity. Now, both of these are stochastic, right? It's just that like these, we know exactly what the statistics behind is here. And this piece here is complicated. And that's why in the paper, I use Gaussian mixture model. But when I'm applying this to an ecological model, I'm going to get whatever the distribution that you get from that ecological model.
Can I ask a very general question? So I'm a bit confused by something basic, which is that I would be shocked if most of these statistic techniques were not known for hundreds of years already. So there are books on statistical analysis, probability, etc. So can you comment a bit on how your recent works, when you say I did a manuscript on this probability that people in biology were not using, were you using things that were well understood and translating them so that biologists could understand? What exactly is going on? Can you tell us a bit about how your works fit with the existing literature?
Sure. Well, the first one is what I already said, like, this piece was not being used in the experiments on plate counting. Now, there are a few challenges that I was talking about, for example, when fitting to the Gaussian mixture model, is that your calculations got really big and you could not come up with an analytic form for the maximum. I think that this was able to be done now. It was because we now have some tools like AutoGrad, like GPU parallelization, because these need to be summed over all possible values of n. So like you usually have to put the cutoff, and I put in the cutoff like 3 times the biggest number that you get from automatically from doing the naive thing, right? Just making the multiplication. Now, another thing, and I think that like henceforward is the most important, is that like models for these, many times they get something that you cannot come up with an analytical representation. So even though there are people who studied a lot, this part is a little bit more complicated. And that comes back to what I was presenting yesterday. Many times you can simulate a model forward, but you don't have a way to come up with the likelihood, right? So this depends on how complex your model is. Now, many people undersimplify the issue, and that's what I was talking about yesterday.
I'm just confused about, of course, there are many people that undersimplify things and don't do things properly. Of course, if you write for a biologist just to multiply by 5 for some biology, it's already an effort. So, but that's fine. I mean, not everyone is doing quantitative stuff, but counting things is very important. There are many people that count things properly and that know how to use statistics properly. So are you saying that until now, people did not know how to count bacteria properly?
No,
Generally, or some biology groups did not know how to do it?
Yeah, some biology groups, and actually many, were attributing to the variability that you get in your case to something biological, to saying that like your probability of n has the same dispersion. And by then they could not differentiate between the noise by the dilution and subprocessing from the saying, oh, is this heterogeneity, is this multimodality actually coming from the underlying ecology?
And probably there are other groups, however, that do a proper analysis, right? So can you comment on your interplay between how your work fits within the community of people that do a more complete analysis?
Yeah, there are many people who do only this part. Do that before. When I was doing, when I was going through this, absolutely nobody had a tool to come up with this part of the problem. On plate counting, specifically.
Okay, thank you.
All right, do we have further questions? I can move to the next topic. Now, there is another thing that I want to do when I get to professor position, which is, I mentioned yesterday, time series forecasting. And molecular dynamics. So throughout the studies of biophysics, they are usually done based on what we call molecular dynamics. And basically what we have is like, you have a full protein that is going to be quite complicated. You think about every atom in that protein, and you create some kind of spin glass system that is going to say, throughout the chemical bonds in that, let's say that I create a potential for each pairwise interaction. The goal then it is to say, now there's a problem in classical mechanics, right? So you just create your equation of motion for the position of every atom in the system. Nice, you have a differential equation for every atom in the system. Now, the way that people usually treat that, it is to say, I create these and then I use all of this complicated ferramental in all of this parallelization to solve this differential equation. By the time that I do that,
can I ask
yes.
a basic question? In this kind of game, the differential equation that you are writing are, let's say, deterministic, or you have some stochastic piece?
Okay, interesting. usually in molecular dynamics, what people do is, what do you usually call the stochasticity? You think about, like, molecules of the solvent, correct? Mm-hmm. So what they do is, like, they put together with your protein, let's say, a bunch of molecules of water, and you also make an interaction between the atoms of your protein and the atoms of water outside. That's what we call atom molecular dynamics. Now, in other cases, people also introduce, like, we can create, like, instead of thinking about every atom of water, you can create, like, some stochastic bump that is supposed to be the atom. Yeah, exactly. So there are people who do either, and that's actually part of the point.
Okay.
Yeah. Part of the point is that, like, by the time that you have all of these solvent molecules, right? Even though this is a deterministic equation, it's going to be highly stochastic on how every interaction happens, right? So what most people treat this, it is to say, I want to find the equilibrium distribution, right? You go from these MZ solutions to equilibrium distribution. So it's very hard by the time that you have the solution for every atom in the system, including the ones that are not of your protein, including the ones that you are solvent, to actually have something meaningful. So many people are going to say, I'm going to have one particularly variable. Some people call these reaction coordinates. Let's say I have the pieces of my protein. I want to know if it's folding. What is the distance between these two separate pieces of my protein? And then you create a plot of these through time, x, and you come over all together. And then people come from these and try to infer a effective energy landscape, right? So for every this variable x is, you're going to have, like, a V of x. So what is the total potential? And let's say if you have a protein that folds and unfolds, this total potential is going to look something like this, something like a double L potential. Now, this is very good. This gives you a series of important results for understanding biophysics, and more important, this is very well physically motivated, right? Even though, to be fair, the physics of this can also be a little wiki, because not only, as Dario was asking, sometimes you have to put all atoms, sometimes you have to put the solvent, you have to check up for temperature, but people can create coarse-grained models, right? If you don't want to write the full differential equation, you go through a simpler differential equation with stochastic terms. And sometimes, even more important, you have to decide if your interaction is going to follow a fully classical mechanics or quantum mechanics. And some examples, you do need to put in the quantum mechanics on it. One classical example is the behavior of chlorophyll, right? So the thing that we learn in school, that is how plants do photosynthesis, actually, the explanation of that was necessary to include the quantum effects. So people do all of this complicated, and then they just try to come up with a potential landscape. Now, my goal is to say, let's go beyond that, right? So what if I try to learn from all of this MV, I'm going to come up with many trajectories from the same system, and I want to learn what is the effective dynamics of the reaction coordinate. So the idea here is, you are not only going to come up with the equilibrium, you're going to say, if I'm observing a trajectory in this reaction coordinate time like this, can I continue this trajectory? Now, as of now, I only saw two papers in the whole literature that try to do this on the time series forecasting. But both of them, they do exactly what I have drawn here, right? You have a past, and then you try to continue with the future. Now, the neural network that is going to give you the future is trained over all of these, all of the trajectories that you have available. Now, my argument is that this is not enough, because as Dario was mentioning, there is these effects that are stochastic. Even though you might be treating every single molecule of the solvent, these methods are still stochastic. So from here, I'm going to have to create not only a prediction of the future, but also confidence intervals. Think about this just for a Brownian motion, right? A particle in Brownian motion. An MD is far more complicated than a particle in Brownian motion. And on that, you're not going to only predict, you also want to know, based on your diffusion coefficient, how far spread your particle is. Does that make sense?
But so essentially, you are saying, following a little bit, if you want, the point that Pedro was pointing. You say that up to now, people just, let's say, throw a trajectory without committing at all about the possible errors that could be large, could be small, and instead you want to, let's say, make serious this statement and quantify how good or bad can be the prediction.
Exactly.
I see.
Yes. And I think that from this, we can come up with effective dynamics for MD, or for the whatever reaction coordinate you are trying to study. Now, on that sense, we need to not only be able to do that, we need to be able to verify through that. And the way that people usually do in machine learning it is, I create my trajectory, and let's say that I create my 50% confidence interval, right? So I'm going to split all of my trajectories into all trajectories into a training.
I'm sorry, Pedro. I find it again very hard to understand these statements that when you use words like people and stuff like this. So is it fair to say that in neighboring fields or in very similar questions, of course, these things were studied? I mean, in active matter, in weather, in stock market, etc. I mean,
yes,
people
exactly.
all the time put uncertainties on predicting time evolution. Of course, again, in very biological fields, exactly the same mathematics might not have been applied yet for some problems. But can you please distinguish a little bit what you are borrowing from something that's very well understood and what you are developing new technology to a new conceptual understanding, just so we better understand
Of course.
exactly what's the value? Is the value in the biology, in the probability, in the machine
Well,
learning? What is the most interesting stuff in all this that we should focus on?
Well, what I am developing new is like creating these systems that we are actually going to come up with the uncertainty quantification. Now, this is based on many different advances in AI. I was talking about yesterday about diffusion models and normalizing flows. And on that sense, I think that this is going to create us a better idea on how the molecular dynamics evolution is. So on that sense, it is now I used to have just a prediction that we could not trust. Now I have something that takes into account the physics and the uncertainty related to that physics. And what we get now, it is on the biology. It should say like, oh, you think about how do you study the reaction coordinates for your protein. Now I'm going to create what is the new dynamics for only these two things, considering everything around, considering both the rest of the protein and the solvent outside.
Sorry, I'm a little bit confused about the use of uncertainty in relation to a molecular dynamics simulation. In principle, molecular dynamics is an exact procedure. Of course, it discretizes the equations of motion, but these are in some sense systematic and not random errors.
Yes.
So I'm not sure I would describe them as uncertainty. I mean, I think if I talk to a professional molecular dynamicist, they would say, I know what the trajectory was for 100 picoseconds.
Yes, but the point is like depending on what your initial conditions is, right? You're going to have for the same system is slightly different, and they are not part of your protein. It might be just from the stochasticity of coming back there. Now, it's just like when you think about Brownian
But the
motion,
actual
right?
trajectory I observed, right? I mean,
Yes.
I could say the same thing with respect to Brownian motion, right? There's the problem of inferring the force field in which a Brownian particle is moving, but I could still imagine that I observed the trajectory exactly, and the
Yes.
uncertainty is not about, right? So
Maybe
uncertainty
the
is
way
related to, is only when I try to infer something from it, right? I don't have any uncertainty about the trajectory itself.
Yes. So the point is like when you have like a particle and then you're going to have like multiple molecules of water hitting the particle. If you know the position of every molecule of water, you know exactly what the trajectory is going to look like. That's correct. And if you are observing it directly, which is something that you do in molecular dynamics, you're going to observe it evolving in time. 1 second. You're going to observe it evolving in time, and then you can in principle know, right, every point that the particle is. But what you want to know, if you want to learn the force field, for example, right, what you want to do is like, no, I want to see it here, and then at a later time, I'm going to come up with a probability distribution from that. And to learn the force field, I need a probability distribution at a later time. This, indirectly, is just like learning the effective force field, right? So I have to learn, I have here the force field, I have here the stochasticity due to diffusion, due to the Brownian diffusion. Now, I want to do exactly that for molecular dynamics. Learn the force field and learn the stochasticity. Does that answer your question, William?
I think maybe, yeah, you should go on.
Yeah, so I want to learn the effective dynamics, just like we do the force fields in Brownian motion, the effective dynamics for your reactions coordinates in molecular dynamics. So yeah, a summary here it is. First, dealing with, as I was mentioning before, ecological models in all over the place, but in specifically in biofilm formation. But every model in more open hand, every model for which P of n given theta cannot be obtained analytically, and on the molecular dynamics, the effective is stochastic dynamics of reaction coordinates. So, as I mentioned here, and I mentioned before, these are just like two trusts that I think are completely possible and will require minimal extra effort, right? These are things that I could pretty much do on my own. Of course, once I get to an institution, many new possibilities for collaboration and visits are going to come up. But these are two projects that I believe I can do it on my own completely once I get there. Any further questions?
I can ask a question, but so is the seminar finished, or is there more that you want
Yes,
to?
it would be finished by now.
Okay, so I guess one question is, how much biology do you need to know in order to get into what you're doing? I mean, is there a lot of biology, or is it?
Yeah, it is on this first one. Yes, there is a lot of biology involved about you creating the right model that represents everything that you have there.
And how do you? So, what can you say a little bit about what the biology that you need to know?
Okay, so for example, you need to my example was to the gut. Let's go back to the gut, right? You need to know, for example, that like, oh, you need to have an idea of what is the rate in which your bacteria connects to the gut. Now, you have to know from the C. elegans, oh, the C. elegans have like some kind of part that is first grinding. So when it eats a bacteria, it's very unlikely that it's going to attach to the gut. That kind of biology, but it's not, maybe it's not like so much biology in the sense of like you need to know the details of how this specific living being is. No, depending on the system that you're going to study, you're going to have to learn the specific, how can I say, procedure, the specific dynamics that the data set gives you. Now, on the molecular dynamics, it's more on the sense of chemistry, right? You need to know the chemistry of the biochemistry, let's say, of the thing that you're trying to study and what results are you going to expect from that.
So if you were working with a biologist, what would you talk about?
I will talk about like, do you think, coming back, right? Do you think that this heterogeneity is explained by this model or that other model?
Okay. Thanks.
So maybe a somewhat related question. A lot of times I'm interested in these probabilistic frameworks, not because I'm trying to make a theory or a model, but because I'm trying to extract more information from experiments.
Exactly.
And so maybe you could say something about whether you have established interactions with experimentalists, whether that's something you need to grow.
Yeah, I do have a few interactions with experimentalists. That was another thing that I mentioned yesterday. I have a group of collaborators in University of Alberta who are talking about like some of these models, but for prior assembly. The molecular dynamics part, I have many collaborators, both here. So the people in University of Alberta are Michael Woodside. Here I have Ben Waskan, and that is like more across the world, many people working on molecular dynamics who could come up with the data set. And many of the data sets were already published and available online. So it's something that I can literally go to their paper, go to the link and download. Now, I am already developing my collaborations on the biofilm formation projects. Many people, both here in the US and in Brazil, they take like microfluidic devices where you see the single molecule single bacterium attachment. Here I also have, as I said, the people working on gut microbiota, even though this is kind of a project that like is going to be fading out from here. So yeah, I already have some.
What you could help us with is if I think about trying to do better analysis of experiments on the growth of biofilms.
Yes.
I mean, there are lots of technical things that one can always do better. And that's a good thing to do, right? But do you have a feeling for what's the big question that, as a physicist, you find interesting about the biofilm? I mean, I could be interested in the applications of things that I know how to do, but I could also look at this thing and say, oh, it's amazing because this film does this astonishing thing that I find very surprising as a physicist, and that's the thing I want to understand, and I'm going to use these tools to try and get at it. So do you have an example like that?
Example, and I was mentioning this yesterday, right? One of the things that I did by doing the mathematics more correctly is that many people could not answer before, right? Whether you see like cells going and you see like there are fluorescence there, right? And then you say, oh, this fluorescence comes from this gene that I marked. And then many people would say, oh, that means that this gene is ultimately producing. And what I did, and I was mentioning yesterday, is that it's not exactly like that. Actually, many things were inherited and people were not taking the inheritance incorrectly. Now, on the gut microbiota and on the biofilm formation.
okay, was it important to know that in some deep, I mean, you shouldn't say things that are wrong. So it's good to push to get it right. But was there some conceptual point?
Yes, the stress regulated genes were understood as if they were always active. And what happens is that like it's only active for a very short fraction of time. At least on that one model is that we are working on, right? So they thought that like you need a very high activation to observe, for example, the fact that they stop growing. And by doing the cell division correctly, I said, no, actually, you only had your stress regulation for a very short period of time.
And that means that other processes inside the cell must sort of carry a memory of that short event. Or I mean, I'm sort of trying to understand.
Yeah, exactly. Like it might be that like, well, from that, it might be that like you cannot just take into account only the single cell. You have like a history of something that happens generations ago that might still be affecting the way that you grow right now.
Okay.
And yeah, on this case, again, is the point that like I want to also quantify, like coming back to the biofilm, you want to quantify how fast they can grow, right? And that takes into, you have to take into account both the small numbers and the large numbers. Now through the mathematics of this, that gets quite complicated because you don't have a single method that works on both regimes, right? But then what you have to create, and you have to create all of the statistics beneath it. Now, I think that part of the point that I was trying to bring also is the first part of my talk, and I'm sorry if that didn't get many close, is that, let's say, I do my experiments and then I attribute my multimodality to the fact that you have like a seasonal pattern, right? I want to know if water is safe. Then you count the bacteria there. It might be that like you observe the two, and that's like, oh, this is a seasonality effect. So like it might be that like as you pick them up early in the day and later in the day, there is some kind of oscillation happening. Actually, if we have for the C. elegans, one of the future goals that we are developing here, it is to say, what if there are two kinds of bacteria and they are competing or even predating inside of the microbiome gut? Can I find evidence for this multimodality is coming from this interaction? Can I see this interaction from indirect experiments like plate counting?
Good.
So yeah, let's say, as I was talking about the model, oh, okay, what if my model includes non-trivial ecological interaction, includes competition, includes predation? And that's something that like if you only do the naive way, you're probably not going to be, because sometimes you're going to miss your multimodality.
Okay, thank you.
Are there any questions from other people?
Well, I was going to ask, but probably he already more or less replied. So, as far as I understand, for the biology part, let's say you need the proper environment somehow, because you are more like you are more a physicist than so do you already know that here in, let's say, in Brazil and in Sao Paulo there is the right environment for that part, right? If I understood from your answer.
Yes, I have connections also in the biology department at USP. I don't know if I told you that I was part of an interdisciplinary program. I don't know, the molecular sciences program. So I do have access to many
Okay.
people. That's how I know people in Brazil
Okay,
who do
great.
this kind of experiment. Yeah.
Okay, great.
Any other questions? Okay. Okay, so thanks very much, Pedro.
Thank you.
Thank you.
Thank you.
So, as I said, probably within a month we'll have some kind of news.
And looking forward to hearing from you. Thank you very much.
Thanks very much, Pedro.
Bye bye.
Bye-bye.
Thank you, Pedro. Bye.
Bye-bye.
Bye.